with us Dr. Kutubuddin.
I would like to interrupt you, sir. Dean of School of Biotechnology from KIT University.
Yeah,
Srinivas, yes, sir. Srinivas sir was there. You can see the face.
Acha. Okay, no problem. Sir, we are glad to receive you, Dr. Srinivas Patnaik.
Thank you, sir.
Okay, sir. Thank you, sir. It is a major setback, the students are entering every moment. Before we start the program, let me introduce each and everybody first. We have with us Mr. David. We have with us Dr. Kutubuddin, scientist, CRRI, Cuttack, Odisha. And now with us, Dr. Srinivas Patnaik, Dean, School of Biotechnology, KIT University, Bhubaneswar. And with us, our dear students from the Department of Biotechnology, KIT Bhubaneswar. This is a special program, all are not invited, very specific students have been invited to interact with Dr. Kutubuddin because, this is a beautiful chance on behalf of US Consulate General, India, Hyderabad and Regional Science Center, Bhubaneswar. We have tried our best actually to engage this platform to congregate, eminent personalities all over the world together to learn together. Now, before we start our program, actually briefly I want to introduce Dr. Kutubuddin. Whatever knowledge I have gathered regarding him, he is a very talented young scientist of India and has been recognized by US also. And he did his PhD from University of Calcutta, and after that he has received beautiful scholarship from US government, and he has done research also there, as I came to know just now. Mr. David was talking with him, and he has also served our country, and especially now he has been teaching and doing research in this field, also in the field of rice production. As far as common man is concerned in the field of biotechnology, we know genes somehow. But today we will request Dr. Kutubuddin to start with the program and to encourage our students first, the way he started his career and the way he was motivated to the field of research, and exactly speaking, the way he has been working in the field of genetic engineering and especially in the field of paddy cultivation. So I request Dr. Kutubuddin to take over the session and start as per your planning, sir.
Before that, sir, can I just request David to say a few words so that Dave has another program to go and attend. So maybe then, if you don't mind,
it will be best, sir.
Yeah, thank you.
Thank you,
Mr. David.
Just a couple of quick words of appreciation first for Dr. Kutubuddin and for the Regional Science Center Bhubaneswar for inviting us for this program today. It's an honor for the U.S. Consulate General in Hyderabad to participate and to kind of bring these two groups together. And we just hope that Dr. Kutubuddin and his experiences can be a good role model for all of these young students and scientists to continue to pursue education both here at home in India and taking advantage of the opportunities that exist outside of India as well. And we're very proud of the Fulbright Nehru program that provides scholarships for aspiring scholars and scientists from India to go study in the United States, not just to receive more education, but we really look to people like Dr. Kutubuddin to be a bridge between our two countries. to be a personal connection as he spent time there in Pennsylvania, which is my home state. So I'm very proud that we have that connection, and I hope that he has made some very important lifelong friends there, that he will invite those same friends to come over to India to do research and to visit and to help make stronger connections between our two peoples. And this year we are celebrating the 75th program. And so we're honored to participate in programs like this one today to celebrate the achievements of this program and specifically the individuals that make it possible, such as Dr. Kutubuddin. So thanks to the Regional Science Center. Back to you, sir, for the rest of the program.
So in this junction, actually, Dr. Kutubuddin, may I request Dr. Srinivas Patnaik to give a brief introduction on this program.
Thank you very much, sir. It's my privilege to say a few words in this occasion. So good afternoon to all. I congratulate Regional Science Center, Bhubaneswar, and US Consulate General, Hyderabad, and US India Educational Foundation to come together to host this webinar on very important topic, that is genome editing, what, how, and why. I welcome all the esteemed dignitaries, Mr. Dave Moyer, Professor T.K. Adhyay, Sri Somen Ghosh, and today's speaker, Dr. Kutubuddin Ali. In 2013, we had Oliver Smithies, who is one of the pioneer in this technology of genome editing. He visited Kitch University and he spoke to all our students during that particular time when he visited for a conference. So we have these kind of very eminent personalities visiting our universities and talking to our students. It is a very regular process. So these kind of platforms basically enlighten and encourage our students to go for further studies and further research programs in India and abroad. So it's, this particular technology, genome editing, has very versatile kind of applications. It has applications in human diseases, it has applications in plant molecular biology, animal models, and crucial and scientific program have also performed on CRISPR-Cas models to target even COVID-19. There was a call from ICMR also in this regard. So this is a very, very powerful tool to kind of tackle any of the scientific challenges that we face in today's time. So being a researcher in cancer biology, I also tried to do this kind of genome editing using knockout mice model, as many of our students and many of the participants, they know about it. However, the beauty of this genome editing technology is rather than knocking out one or two genes, you can knock down multiple genes, multiple targets you can inactivate. That is the beauty of this genome editing. So at the end, this kind of platforms enable students and researchers to choose and pinpoint their research career and research collaborations. I really look forward to the talk, and I'm sure all participants will be greatly benefited and motivated by the talk of Dr. Kutubuddin. Thank you so much.
Now, thank you, Dr. Srinivas, sir. And we are very happy to listen to you and got motivated our students. And now I request Dr. Kutubuddin to take over the session, sir.
Thank you. Thank you, RSC Bhubaneswar, for inviting me. And good afternoon to all of you, Mr. David Moore from US Consulate General, Professor Srinivas Patnaik from KIT, and Monica Setia, who actually contacted me first. And we decided to talk about something else because we thought that it would be for school children. But later I came to know that it would be for the biotechnology student from KIT. So then I switched the topic because a lot of people are talking about genome editing, and I think biotechnology student from KIT, this topic would be very suitable and timely. So, and I tried to prepare my slide very in a simplified manner so that every student can get it. And also I should mention before starting my talk, so whatever I I was exposed to this wonderful technology through Fulbright Fellowship. So I wrote a project on this technology, and I went to USA for doing work on genome editing, and that was my first exposure to this technology. And then I came back, and I'm continuing on the working on this technology for rice improvement and some other very interesting crop improvement applications. So I would share all of these things through my slides, as it is an awareness or public lecture type of thing. So I would be citing a lot of others work, also not only highlighting my works, but there are some slides where I showed my work also. So let me share my screen here. Okay, so is it visible clearly? Is it okay?
Yes.
Okay, thank you very much. Okay, so thank you again. So yeah, so genome editing, as highlighted by Professor Patnaik, is a wonderful technology, tremendous potential in impacting agriculture, basic biology, and also human therapeutics, like human medicine, you know. So, you know, let me just share a story of wheat. Wheat, a human being are dependent on wheat as one of the principal food. But many people are allergic to wheat. They cannot have wheat because they have problem whenever they have food containing wheat. Happens because this wheat has a protein which is called gluten. And that's why this allergy is commonly known as gluten allergy. So this gluten protein is nothing but a mixture of two different proteins. One is gliadin protein, another is glutenin protein. Now, those people having gluten allergy, they consider this gliadin protein fraction as a bad protein. So they start immune response against this protein. And as a result of continuous immune response against this protein, the people, they suffer from celiac disease. So what happens in celiac disease? Whenever they have food containing any wheat protein or wheat component or rye or barley component, they have severe stomach pain. Because of this continuous immune response against this protein, they are, you know, this is a small intestine, human intestine, and these finger-like projections are called villi, and they are responsible for absorbing nutrition. Now, due to this continuous immune response, because their body treated this gliadin protein as a bad protein, their villi become destroyed and they are very less efficient in absorbing nutrition. So this is a real problem. So people cannot have any food which are made up of wheat, barley, or rye. So how to solve this problem? So the question is here, is there a way to disrupt specifically the gliadin producing elements in wheat? So this protein is produced from something. So can I disrupt, or can we disrupt specifically that element so that gliadin protein does not produce? This is my question to you now. So the way is genome editing. After the innovation of these tools, we can do that, specifically disrupt the element so that it cannot produce the gliadin protein. Okay. So, you know, suppose what I showed you, a chain of letters, you know, A, G, C, and T in red color, in yellow, green, and blue color. So suppose this is the code language for producing the gliadin protein. Now, if you do genome editing, if I want to give you an analogy, so this is a text. I am functional. Now, if someone edit that text and add a non before functional, then it becomes I am non-functional. So if you edit the text, then the meaning of the text changed. Similarly, if you edit the genetic code here shown, also because of the changed code, it would not be functional as it was doing in its original form. Okay, so this is called editing or genome editing. So now this is, you know, Jennifer Doudna and Emmanuel Charpentier, who got Nobel Prize this 2020 October last year. for developing and inventing a method of genome editing that's called CRISPR Cas9. And they published their first paper in 2012, and it took only 8 years to get this Nobel Prize. So this is one of the fastest event in the Nobel history, that in between 8 years they got Nobel Prize after their first publication in scientific journal. So this is really a tremendous technology that has huge, wide array of impact in human society, and that is why Swedish Academy of Sciences decided to award them. So I will be talking a lot about this technology and how this technology has been developed so rapidly. Okay, so let me give you the outlines of my today's talk. So in part one, I will be talking about what is genome editing. So partly I already talked about. And part two, I will be talking about how we would do genome editing. So brief mechanistic briefing I will be giving you, but that would be in a very simplistic manner, not the technical glitches. In part three, I will be talking about why we need genome editing. So why we need to do genome editing. So in order to understand genome editing, we have to know what is genome. So as a human being, our body made up of trillion of cells, and each cell has genetic information, and a complete set of genetic information that is present in each single cell is called a genome. So if I want a more simplified definition I want to give you, here you can see genome is the entire DNA sequence of an organism that is present within a cell. The genome is essentially a huge set of instructions for making individual parts of a cell and the whole organism, and directing how everything should run. So from this definition, you should understand that genome actually, directly or indirectly, controls almost everything in a living organism. So if I want to go into deep or further molecular details of this genome, as I told you, human cells are made up of trillion of cells. Similarly, higher plants, like here I gave you example, rice, these are all made up of trillion of cells. And if I want to give you an example of plant cell, this is a plant cell, and this is the nucleus. All of us probably know this because we all had biology in our schooling, okay? So this nucleus is the head of this plant cell. Why it is head? Because nucleus has chromosome in it. So this chromosome is the hereditary material that actually passes from one generation to another generation, and this chromosome is nothing but made up of long stretch of DNA. And this DNA, but long stretch of DNA, obviously this is true, but the whole stretch of DNA are not equally functional. Some part are functional. Suppose here I highlighted with the green box, and this functional unit of a DNA stretch is called a gene. So in a long stretch of DNA, there are several functional units, and every functional unit is called a gene. So now why it is functional? Because this gene, when it expresses, then it produces some protein, and this protein governs the character of that organism. Suppose somebody has a black hair, somebody has a brown hair. So that is also governed by some kind of protein, and that protein is produced from some kind of gene. Okay, so this is a flow from gene to protein, and then protein actually governs the character. So now what is there in our gene? If you see in our gene in molecular details, there are only 4 letters, A, G, C, and T. These 4 DNA letters actually makes up our genetic code, and this code controls everything. So now if I want to give you a virtual example, a real example of a DNA sequence, how it looks like. So this is a stretch of DNA sequence, and you can see there's a beads of DNA letter, only 4 letters, A, T, G, and C. You cannot find any other letter there. So these 4 letters with different combination, they make different gene. Gene 1 has the same 4 letter, gene 2 has the same 4 letter, but this 2 gene has different sequence. Okay? So coming back to that problem that I introduced you in my first slide, the gluten allergy problem of wheat. So if I want to show, this is the gliadin gene that produces this gliadin protein, this red ball-shaped gliadin protein, after it expresses. Now, the question is, we have to disrupt this. As I told you, the disruption mechanism is genome editing. We could do with that tool. But the question is, wheat plant has more than 164,000 genes. Now the further problem arises how to pinpoint gliadin gene. If I want to go to disrupt gliadin gene, is it also possible to disrupt other gene? So how to pinpoint that gliadin gene? So this CRISPR-Cas9 that was developed by Emmanuel Charpentier and Jennifer Doudna is actually because this technology is so powerful that you could pinpoint that gene that is of your interest. Now, coming to part 2. So how we do genome editing and how these tools actually function to detect a specific gene and then disrupt that gene. So if I want to give you an analogy, this tool, CRISPR-Cas9, is actually like a scissor, and that is a great tool for targeted inactivation of a gene that is a functional unit of DNA. So this tool has two different part or component. One is a customizable RNA. A customizable RNA means this is a small RNA and you could customize as per your need. And this is another component is a DNA cutting protein, which is a Cas9 protein. If you want to give you a simple analogy of these two components, this Cas9 protein is a postman, and this postman has two scissors in its pocket. And then this customizable RNA is a genetic address. So when you give this genetic address to this postman takes the address, and then it forms a complex, and it goes to the genome, and it finds for similar sequences. Because this RNA you designed, you gave the address, and whenever it finds similar DNA letter in the genome, then it binds there, and then it gets a signal. So this is the address where I need to make a cut. Okay? So then after it gets signal, then it utilizes two scissors. One scissor cuts one strand of DNA, another scissor cuts another strand of DNA. So that is how you can make a very precise and targeted double strand break in the DNA molecule that you want. Now, it is clear that it could be used to cut a DNA of your interest, cut a gene of your interest. But I have not talked about how it actually helps in genome editing. Whenever there is a double strand break or DNA break in cell, cell takes it very seriously because it is not common and DNA is genetic material. So any changes or any damage in DNA is a serious matter for cell. So then what it does, it immediately activates this repair mechanism. It has to repair this kind of damage. So it employs its repair mechanisms like NHEJ, non-homologous end joining. This is the predominant pathway by which a cell repair this break. Now, but this NHEJ, this is a kind of error prone. In 90% cases, it repairs the break, seal the two broken end perfectly. But in 10% cases, it makes an error. And due to this kind of error, it randomly insert or delete some original letter or introduce some new letters. So here I have shown in red color. So due to this insertion or deletion, means new letters or original letter deleted, the whole sequence of that gene is disrupted. And whenever the sequence is disrupted, then this gene would not be producing the same protein as it was producing in its original form. As I told you, sequence is everything. Letters are all same, but sequence is everything. So because of this insertion and deletion, the sequence is disrupted and this gene function is gone because of this sequence disruption. So from this video, you'll be understanding more clearly how it actually works.
Been adapted into a powerful tool for genomic research. There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, I think it's more clear to you. And so coming back to the same story of gluten, as I introduced you to that problem, so this has been already, CRISPR-Cas9 has already been used to get rid of that problem. You know, this is the gliadin gene I showed you, and this is the portion from where the antigenic property actually arises. So this region need to be deleted or somehow disrupted. So what they have done, they designed two different genomic address. Here you can see this arrow, blue color arrow, and they made double cut from here and here. And what they have seen different kind of genotypes arise. Here you can see one nucleotide or one DNA letter deleted, another DNA letter deleted, here 3 letter deleted. So 1 dash means 1 letter deleted. So there is a lot of different kind of disruption happened. And when they have seen the phenotype of this one, when they have, so this is a spectrophotometric analysis where they have seen, here you can see this graph showed the presence of alpha gliadin high amount in normal weight. But here this one, the edited one, the mutated one, that has a very negligible amount of that alpha gliadin due to this kind of gene disruption. And if you see from outside how it looks like, the edited wheat, and this is the control wheat, they're all similar. So 1 second, okay. So you can see this is edited and this is control. They're all similar. The only difference is that the edited has a very negligible amount of alpha gliadin compared to the one which has not been treated. Okay, so coming to a different dimension from this normal technology that could be used for disrupting any negatively impacting gene. Suppose you know this gene is negative for a crop, this gene is negative for an organism, so you could disrupt that with that one. But normal CRISPR-Cas9 cannot be used for kind of correction, you know. So this is a tradition. This is an example I often used in my presentation. So this is a Oro from a movie Pa. So Oro is 12 years old and his friend is also 12 years old. But you can see the difference between them, how they look like. Oro looks like 80 years old. So this is because Oro was suffering from a rare genetic disease that is called progeria. And, progeria causes rapid aging in childhood. And the affected child looks like very aged personality, and they die in between 13 to 15 years old. And Oro has already also died because of that disease. You know why they look different? Because in one of the gene, they have this simple difference. This healthy girl has a G DNA letter, whereas Oro, instead of having A letter, because of the single wrong letter inserted in Oro's gene, Oro is suffering from progeria. So we had all those kind of information, but why progeria happens, but we did not have any tools. Now, in a plant perspective, this is a Kitake rice plant, and this is a mutant one. So the difference between these two genotypically, this has a C DNA letter, whereas C is replaced with a T letter, then it becomes resistant to a fungal disease. And you can see this one is susceptible and died due to the fungal infection. So here you can see, so obviously this question arises that is there a way to rewrite genetic letters? Precise rewriting, not randomly disrupting. Precise rewriting is needed to cure Oro or to make a plant disease resistance. Okay. Now this technology is called base editing, which is a sister technology of CRISPR-Cas9. This is derived technology from CRISPR-Cas9, which act as a pencil, and it has the ability to rewrite genetic letters from A to G, vice versa, T2C, vice versa, and also C2G. And this has been pioneered by David Liu from MIT. So, let me give you a simple example of how this base editing works. So, this is, I don't want to read it, so you read it, what is written here. And this is the sky without any star, okay? Now, if you want to correct it, you have to apply base editing. And once base editing is applied, then this aim changes to an AS and it becomes functional, and the sky is full of star now. So this is how base editing works. So single genetic letter or single DNA letter, if it is wrongly inserted, then it could be corrected with base editing. Now, this is another small video too.
Those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay, great. So, okay, let me give you an example. Is it really happens in plant or animals? So this is an example from my work, what I did during my Fulbright, and also continuing here in India. So this is how we actually determine what kind of sequence is there in which gene. So this is called Sanger sequencing technology, and this is a peak. If there is a blue peak, so this is an indication of C letter. Green peak means A letter, black peak means G, and red means T letter. So this is a snapshot of the gene sequence of WSL5, and we targeted this A DNA letter to change it to a G letter. With the application of base editor, we could achieve specifically changing A letter to a G letter. You can see this green peak has been changed to a black peak, which is an indication that A genetic letter has been converted to a G genetic letter very precisely. And you can match other peaks around this G. Those are all similar with the control one, which is not treated, and this is the one which we have obtained after treating our plant with the base editor. So those are not changed, but only the A, target A, has been changed to a G. And because of this single nucleotide change, as expected, we have found this kind of phenotype, which is called WSL5, means white striped leaf phenotype. So this is how single nucleotide has been changed with base editor for a phenotype. So for another example, for another gene, what we had achieved similarly, in this case, this A was our target, and we also achieved a similar result. A has been converted to a G nucleotide with base editor, and similarly Z3 phenotype we have observed. So you can see this one is not treated with this base editor, but these are treated with base editor on which have severe phenotypic impact. So Z3 means Jevra3 phenotype. Means it looks the leaves of that plant looks zebra kind of thing. So light green, dark green, light green, dark green, light green, dark green patches, and that's why it is named Zebra3. And this Zebra3 phenotype arises from the single nucleotide change in that gene, but other sequence remain unchanged without any disruption. So coming to this part 3, as I have mentioned, sorry for disruption. मेरा एक presentation हो रहा है अभी नहीं चाहिए मैं बात कर रहा हूँ please. Hello. Sorry for the disruption, as someone was knocking in my door. Okay, so coming to the part 3. So now why we need genome editing? So I would be trying to validate this point that this has a huge impact. This could be very impactful in all the arena of human life, starting from food, basic research, and also therapeutics. So let me give you a simple example of rice pathogenesis. So whenever there is a bacteria called Xanthomonas rice bacteria, when that infects to rice plant, then these bacteria actually utilize rice, the genes from rice itself. So how it utilizes? So here is the story. There is a gene called sweet gene. This gene produces a protein which is called sweet protein. And this protein is a transporter protein, is a pump protein. This protein pumps sucrose from inside to outside, from cytoplasm to apoplastic region. So whenever this bacteria infect, zoobacteria infects into the rice cell, then this zoo secretes a protein which is called TAL protein. This TAL protein goes and binds with a nearby sequence of the sweet gene. And this binding induces the expression of that gene. Means this binding produces more protein out of that gene. Okay? So you can see here from this cartoon, there are a lot of protein produced because of this binding. And as there are a lot of protein produced, so it is very rational to understand that they will pump more sucrose from inside to outside. And whenever there is more sucrose outside, then bacteria is very happy because bacteria has the nutrition. Bacteria can use sucrose for their own nutrition, and they will multiply more, and they will be able to infect rice plant in a more vigorous manner. So this is how bacteria infect rice. So they utilize rice gene for their own benefit. Now we know this fact because of the tremendous basic research. Now, if you see in further insights of this one, so this TAL effector binds with a special sequence of that gene, SWEET11, and this sequence looks like the simple sequence. This is called elicitor binding element. But this protein coming from the bacteria does not bind anywhere. It binds very specifically to this EBE sequence. So the question arises here, can we specifically disrupt the EBE sequence? If we can disrupt the EBE sequence, as I said, this TAL effector is very specific. It would not be able to bind. And if it does not bind, then the sweet protein would not be produced in more amount. So the bacteria would not be benefited from the sucrose. Okay? So the plant would be resistant. So this is a simple hypothesis that I told you, and this has been used and already demonstrated in many studies. You can see here, after CRISPR-Cas9 application, this EBE sequence has been disrupted. They have deleted 4 or 5 genetic letters in the treated plant, and as a result, they have got this kind of phenomena. You can see this one is the one that has not been treated with CRISPR-Cas9, that has not been edited. But these 3 panels, they have been edited. And you can see these non-edited plant and normal wild type plant, they are hugely infected with bacteria. But these 3 are very less infected or very minimally infected. So they become resistant against this bacteria. So this CRISPR-Cas9 is becoming a boon to many plant pathologists to develop disease resistance against many pathogens. Okay, so coming to another part, yield. So I'm not going into details. So this has already been used to disrupt many genes that actually negatively impact the production of paddy or rice. So if you could get rid of that negative impacting gene, so you could get more benefit and more production out of that. So you can see this is a normal plant which has not been edited, but these are edited plant which has a vigorous growth and more number of panicles, and they have significantly more number of grains in that plants. Okay. So coming to another very interesting story, you know, what we eat today is modern day corn, and these modern day corn has been evolved from teosinte, taking 9,000 years by some natural selection and then artificial selection. And now you can see this, the ancestor teosinte had only few, means 12 to 15 or maybe 10 kernel or 10 grains in each cob. But here you can see thousands of grain in a single cob. So that is how it impacted in our food security. Now, but it took 9,000 years. Can we do it quickly? Here is an example. So, you know, there are many genes which is actually called domestication genes that genes are required to make a crop very suitable for harvesting, very suitable for our food consumption and very suitable for growing in a very small area. So there is an example, SP gene, and this is in a wild tomato which grows very vigorously here and there, and it takes a lot of space to grow. But domesticated crop, what we cultivate, that has a unique features that it should be compact in nature. So it could be grown in a very small area, okay? So when this gene was disrupted, you can see these wild tomato became very compact in size. Similarly, if you see this one from wild tomato has few flowers in an inflorescence, but when this multiflora has been edited, then it becomes, means multiple times more florets could be seen in that edited plant. Similarly, if you see the fruit size also, this is the wild type one, the wild tomato. And this, when this FAS8 gene has been edited, the size and the locule number has been increased very significantly. So it could be done within 2 or 3 years using this technology. As Professor Patnaik mentioned, that this technology could be also used simultaneously to knock out different genes, to disrupt different genes. So here is the example. If you could go and get 3 genes disrupted together, so you could get all 3 characters together in a single plant, and that has been already published also. Okay, so coming to an industrial application. So, you know, waxy corn is very lucrative for the industry for processed foods, adhesive, and high gloss paper. But this corn is full of, starch. The starch has 2 components. One is amylose, and another is amylopectin. And if you see the biosynthetic procedure, ADP glucose to this ADP glucose goes to 2 pathways. One is amylose, another is amylopectin. So what they have done, the enzyme that is responsible for synthesizing amylose, this gene they have disrupted, and what they have found, when they disrupted this waxy gene, they have obtained a high amount of amylopectin containing corn plant. And high amylopectin corn is called the waxy corn. So they have achieved this waxy corn development through CRISPR-Cas9 by deleting this waxy gene. Means one part you are blocking one pathway and you are allowing other pathway to occur. So this is Huirang Gao, one of my friend who actually developed this one from Kottaba Agri-Science. So he developed, he gave me this photo. So you can say this is a conventional corn, whereas this one is developed from CRISPR-Cas. You cannot distinguish between them, which one is CRISPR-Cas mediated waxy corn and which one is normal one. So there's another example from, this is example from Pennsylvania State University. So I worked with him, Professor Ian. So, you know, mushroom, when you cut mushroom and keep it for a longer time, then it becomes brown, especially the cut point, because it secretes some phenolics, and because of the oxidation of these phenolics by polyphenol oxidase, it becomes brown color. And this is very negative in terms of marketability, because people do not want to buy those kind of things. So, you know, 60% of US corn is actually grown in Pennsylvania, and they supply to other US states. So what Young's lab and other members have done, they have disrupted this PPO gene in mushroom, and they got non-browning mushroom. This has been a few years ago. Okay, this is another very good example. If anybody asks you, name one crop that has been already commercialized using genome editing technology, this is the example. It's called calino. This is a modified soybean that is almost like olive oil. So it is very healthy because it has 80% oleic acid and 20% less saturated fatty acids. So this is very good for health. So this is a huge calino crop growing in the field in US, and already a supermarket is selling this calino. And this has been developed by Dan Foyt's group in University of Minnesota. Okay, so let me give you one of my example that has not been published, but that is in process. So this is a classical example. If a gene has a sequence, if a gene has this kind of DNA letter TAG in continuation, TAG is a stop codon. It means this is a signal where protein synthesizing machinery should stop. So this is called stop codon, okay? So TAG. Now, because of certain mutation happened in rice, there is a premature stop codon. Premature means it should have been at the end of the gene sequence, but now it is appearing at the middle of the gene sequence. So the whole protein, the whole length of protein could not be produced, and that is why this rice plant becomes disease susceptible. So it has a lot of disease. Now we thought that can we cure it with base editing tools? So we applied base editor and we successfully modified this A into a G. So it becomes TGG, and TGG is no longer a stop signal for the protein synthesizing machinery. It is now a green signal. Okay, so it will continue protein synthesizing until the end of that gene. So now it will produce the full length of protein, and now the plant would be disease resistant. So this we have already achieved for developing disease resistant plant. You know, now, although I don't work on human therapeutics and all these things, but I keep update myself. So I thought it would be good to share some very exciting example, recent example on how it is being applied in human therapeutics. So, you know, this is a very common inherited deafness gene. This is called TMC1 gene, and because of this, of a single wrong letter C instead of a T, T is the correct letter and C is the wrong letter. Because of the C insertion, the affected people, they become deaf. And you can see here is a cochlear cell, outer cochlear cell, inner cochlear cells in ear. So what they have done in a mouse model, which have a recessive deafness, and they applied base editing tool and they corrected C letter to a T letter. When they corrected, you can see this is completely looks like the one which is normal after correction. So the cochlear hair cells has been restored with base editing tools, and the mouse can hear now because of this treatment. So this is another recent one, just May 2021, they have published this one. So, you know, coronary heart disease also, this is very common in many parts of the world. Heart attack and stroke we see, and we often narrate that, oh, I just talked to him and he is no more because of the heart attack and stroke. So this is because of some kind of bad cholesterol. You know, there is high blood, low density lipoprotein. So here is the yellow color one. If it happens, then the vessels become clotted, and the blood cell cannot move. Okay, and because of this kind of problem, they suffer from heart attack or a stroke. So what they have targeted, this gene is well known, this PCSK9 gene, and they have a correction. If it can be corrected from A to G, A letter to a G letter, then this problem could be solved. And they have demonstrated in a monkey that it significantly lowers the cholesterol in primates by applying this technology. So if I want to show you how it actually happens, they have presented this kind of model in human also. So first one, you know, I described here. So here is a lipid nanoparticle where you supply the CRISPR mRNA, and here is the address. So here is the postman and here is the address. Then they go and bind. They go and bind with a specific gene, and they correct that A to G conversion. They do it so that the PCSK9 gene become turned off. And as it is turned off, then the blood cholesterol level becomes minimized. So the risk of heart attack and a stroke also minimized. So if we want to give you an update, what are the treatments that are under clinical trials? So these are some clinical trials that is going on in USA mostly. These two against making cancer, killing T cell, and also some blood disorder, very well known, sickle cell anemia and beta thalassemia. But those are ex vivo editing. Ex vivo editing means you have to isolate their blood and then correct outside by treating with the editing reagent, and then again transfuse them into their body. So those are ex vivo editing. And here is an in vivo editing. In vivo means you have to inject the gene editor directly inside the human body. That is called in vivo editing. And this is for local editing for eye disease, LCA, inherited childhood blindness. These are all different phases of clinical trials in US. So this is a very exciting example of this one success story. Almost 26 or 7 months, maybe 1 year has already been passed. Victoria Gray was the first patient to get treated with CRISPR gene editing treatment, and she is doing very well, and a lot of media coverage is going on around her. This is another very, so for Victoria Gray, you know, this example is ex vivo editing. So her blood was extracted and then corrected and then again transfused. This is ex vivo editing, outside body editing. But this is Patrick Doherty and another 6 patients. They have gone through this treatment that is called in vivo editing. So CRISPR Cas9 has been injected into their blood directly, and 6 patients are doing very well. And these are, this is also under clinical trial, and fast observation after 30 days has been recently published in New England Journal of Medicine. This is also exciting. Okay, so I want to finish with this slide. As I showed you the example of PA movie, the Oro and Progeria, all these things. So this has already been demonstrated in a mice model which had Progeria disease. So, you know, these videos.
So let me pick them up. Hey, honey. Hi. There he is. You see everybody? Say hi. And then we have, as compared to the C57, they still have a very nice sleek coat. Still very, very active. If I were to pick them up, they can jump out of my hand quite literally.
Okay, so now I should stop here. So if you want to know more, you know, I wrote this blog in a very easy way for students in Arjun. You could find if you want to know about more about base editors and want to more easily understand. And if you want to apply these in your experiments, so we edited these 2 volumes, volume 1 and volume 2. This contains many protocols for applying these very wonderful technology to your organism of interest. So you could use those protocols also. And you could also go through this recent review also we wrote on base editor. These are my small group. We are working with close collaboration with other lab also. We are working on different exciting projects on rice improvement, but those are not yet to share because we don't have enough data to share with you. Thank you very much, and I need to acknowledge the funding from NRRI and Indian Council of Agricultural Research. And as I told you at the beginning of my presentation that I have been exposed to this wonderful technology by means of this Fulbright Fellowship from USIEF, United States India Educational Foundation, and I used also the funding from National Science Fund, USA. Thank you very much, and I'd be love to interact with you, or if you have any question or any queries or anything else. Okay.
Actually, students, here is a chance for you to interact with Dr. Kutubuddin and Dr. Srinivas also.
So if you have any kind of question, any basic question, how we introduce those into plant, how we detect those mutations, any kind of queries you could ask me or Patnaik sir also. Yeah. So no problem. No question is silly question. Every question is very important question, you know.
Excuse me, sir.
Yeah.
I'm Anuksha. Can I ask a question?
Yeah, sure. Go ahead.
I'm from fourth year. I'm pursuing biotech, BTech, MTech. Sir, you talked about the stop codon defect. So,
Yeah.
how often is it found in plant varieties?
So, stop codon is present in every gene, but at the end of that gene, at the end of every gene. But in some cases, whenever there is a mutation happens, sometimes it has premature stop codon. So it comes at the beginning or at the middle. So, the gene cannot produce the whole length of protein. So whenever there is some kind of cases, then you could use this tool to correct them. But normally stop codon is present in every gene.
Thank you, sir. That is what I was asking. How often is this defect found in a certain plant?
Yeah, no. I don't have proper statistics to tell you that this much percentage has this kind of premature stop codon. But mutation is common, but nobody knows actually how often it founds. Maybe I can say I only can give you an assumption, but not any definite statistics. And more we do basic research, more we would know that this gene is malfunctional due to this premature stop codon. But there are a lot of cases where many important gene has premature stop codon. But I cannot give you any statistics, but that I should not be given any statistics. I would be wrong there.
Thank you so much.
Sir, this is Devdipta Mukherjee.
Yeah.
Sir, I'm a fourth year student. Sir, when you said we are using CRISPR Cas9 to basically disrupt the gene, and when the body is healing that, it's healing in a wrong way. So basically that is what is allowing us to disrupt the genetic code, right?
Yes.
So, but when we are doing base editing, how are we ensuring the exchange of one nucleotide with the specific nucleotide of our interest?
Okay, that's a good question. I did not go into the details of the methods in my presentation, and that would have been very long. So that is why, there is a protein that is fused with the Cas9, and that protein is a deaminase protein. And that actually Cas9 do the job of transporting that protein to that gene of interest. So you fuse the protein that is called deaminase with Cas9, and then Cas9 takes the protein to your nucleotide of interest, and that protein will change the nucleotide, suppose A to G or C to T. But Cas9 would not do anything. Cas9 only go and bind there and make a small cleavage that is called nickase. Means nick, one single strand nick, not double strand. So I can, it's very, it would be difficult for you to understand if I want to give you methodology without, it's only by narrating. I can share with you a lot of documents for easily understanding. You can go through the plant-based editor at GenBlock, you would be very easily understanding that one. So there I have narrated all the mechanism, how it actually happens. Deaminase protein actually, adenine has a amine group, NH2. So yes. Deaminase protein deaminate that adenine, and whenever adenine is deaminated, then it becomes inosine. And this inosine is treated as a G in cellular environment. And when cell treat inosine as a G, then in the next round of repair and replication, that I became a G. So the initial letter was A, in between I, and then finally it will be G. Okay?
Yes, sir. Thank you very much. So,
yeah.
Sir, is it like base specific? The enzymes used are
yes,
base specific for each separate base.
You're right.
Okay,
for adenine base editor, we have to use adenosine deaminase. For cytosine, we have to use cytosine deaminase. Different deaminase.
Okay. Thank you very much, sir.
Yeah.
Sir, excuse me. I am Shagnik from fourth year as well. I have a question from continuation of Devdipto's question, sir. Sir, my question is that then in this case we are using specific base editors apart from Cas9 in the complex with CRISPR as well as Cas9 as well as a base editor in this case.
Yeah, you are somewhat right, but not exactly right. So base editor is actually CRISPR means the guide RNA, that is the address, and then the Cas9, and then fused with a protein. So these three components, guide RNA, Cas9, nCas9, this is called nCas9, and then fused with a protein. So there are two proteins, Cas9 and deaminase and guide RNA. These three together is called base editor.
Okay, sir. So that means in this case, like you presented in one of the slides that the cells are using NHEJ as one of the techniques of editing the already damaged gene by Cas9. So, sir, in this case, like is NHEJ not working? Only base editor is working.
Yeah, for base editing, NHEJ, in order to work NHEJ, you have to make a DSB, double strand break. But for base editing technology, we don't make a double strand break. We make only single strand break. It's a nick. It is a nick. DNA nick means one single strand break. And this nick is to induce cellular mismatch repair mechanism. So that repair is different from NHEJ. Okay?
Okay, sir. Thank you so much.
Yeah.
Anybody else?
Sir, if it's not an issue, could you, by any chance, share the presentation that you gave us today so that we can go through if it's not a problem?
Yes, I do.
Thank you very much.
Yeah.
There are many. Actually, you can ask. This is a chance. Actually, you can interact with a person of eminence.
Sir, I have one more question. Sir, my question is that like how are we injecting the Cas9 and CRISPR system into the host? Like how are we injecting it? Is it like a injection by a by plasmid vector kind of a thing or something different?
Yes, for plant, what we do agrobacterium mediated transformation of a plasmid vector. You are right. So we do plasmid vector transformation into plant cell, but many group they do RNP transformation, so they don't transfer a plasmid DNA, but they go for Cas9 protein, purified protein, and then the RNA, in vitro synthesized RNA. So protein and RNA they inject. So this is called RNP transformation. So you could do both of them. Means as per your requirement.
Okay, sir. Thank you.
Sir, may I request Dr. Srinivas Patnaik to highlight the present topic of discussion with that of findings in COVID-19 virus, some sort of similarities and linkage. Can you highlight, sir, for 2 minutes? Dr. Patnaik is there or not? Sir, Srinivas, sir. So may I request Dr. Kutubuddin to speak on this topic for some time?
Yeah, I can talk about this. So, for COVID-19, this CRISPR technology also used in USA, especially, and in a very significant amount, because this CRISPR could be also used to develop a diagnostic tool to detect the presence of virus in human sample. So, but that is not a part of genome editing. That's why I have not kept in my slides, but that's a different field. But, because this Cas9 with the specific genetic address means RNA, if you give together, then this Cas9 can find any nucleotide of your interest. So if you design a genetic address or RNA that is similar to the viral RNA, the COVID-19 RNA, then if you isolate samples, suppose nose swab or throat swab from a diseased human being, and it is in the tube, and if you give Cas9 and the RNA that you designed, then even that tube or in that sample, if coronavirus is present or the RNA of coronavirus is present, then Cas9 would be binding to that. And that binding can be detected artificially. And if we see there is a specific binding, and that's an indication that the human sample has coronavirus RNA in his nasal swab or throat swab. And that can certify that this human is COVID positive. So there are mainly two initially developed tool. That is, one is called Detector, another is called Sherlock. These two has been developed using CRISPR technology, and you can Google it. And they has USA has already approved them for detecting COVID virus, and especially in California and Boston area, this has been used for detection from the patient sample also. And India, if I want to say, there is from IGIB, CSR IGIB, they have developed Feluda. One kit is called Feluda kit. That is also based on FN Cas9, that is a different Cas9. And the system has been developed in such a way that if you have a strip, like the pregnancy kit, that kind of a strip, if you dip into the sample, then if there is a double band, then you can say that this is the COVID positive one, and another is a single band which has COVID negative. So this is called CRISPR diagnostic. This is a different area. So you could also explore by searching in Google. There is a lot of resources available. So if you want, I can also share with you. This is the development, yeah. I cannot hear you. Probably.
Yeah, I got. Actually, my question was, the Covaxin developed by Bharat Biotech Lab, was not accepted in the international community for a time being. And what are the reasons behind that?
So there is some protocol that in order to be recognized internationally, you have to publish your finding or study that this Covaxin has been gone through clinical trial in such number of huge mass, and this is the result. There is no adverse effect and all these kind of things, and there should be a result publication in a good international journal, but that was lacking for Covaxin. But they have recently published in a good journal also, so hopefully in future it would be also recognized internationally.
Another question, sir. The Pfizer product, it is 95% efficient, they said. But, while India declared the Covaxin, say, for example, hardly 68% effective. So where is the difference of application of knowledge in that?
So you're saying effectiveness?
Yes, efficacy.
Efficacy. So this efficacy calculation also comes from the clinical phase trial study. So if the study is complete, and there is a systematic way to study how much efficient a vaccine is, okay? So this is a statistical analysis. So how there is a standard protocol that we have to go through for your efficacy analysis of your vaccine. So this is the result of outcome of that study.
Okay, sir. Thank you so much. Is there any student left behind who want to ask a question to sir? Because we are going to close the session now. It is going to be 4:15, and as per our schedule, we have to close 4:15. So this is the last chance for students, please.
Excuse me, sir. I'm Manali. I'm from 4th year. My question is related to CRISPR. Like, what are the scientific uses of CRISPR apart from genome editing?
Okay. Apart from genome editing, this is one thing that I just mentioned. CRISPR diagnostic is not a genome editing thing, but this is coming in a very big way. Also, you can go for live imaging, chromatin imaging. Probably you are biotechnology students, so you have read about fluorescent in situ hybridization fish. So fish is being replaced by CRISPR live imaging. So you can detect in a live cell, you can detect where is your gene, where is your DNA of interest, or even RNA imaging, where is your RNA, and their dynamic movement during cell growth also could be visualized with this CRISPR tool. So that is not an editing, but you can go for DNA or RNA imaging with CRISPR. Another thing is you can also down-regulate or up-regulate gene without disrupting any kind of sequence. So suppose one gene is not getting expressed in a higher amount, but you want that gene to be expressed in higher level, so you could apply CRISPR activator. Another is CRISPR interference, CRISPR-I. So using that, you can down-regulate a gene. Suppose you want to reduce the expression of a gene, you should use the CRISPR-I. So that could also replace RNAi technology. So these are all, but this is not limited. A lot of technology is being derived from this. Even targeted sequencing also is being done using CRISPR technology. So huge kind of application is being done. So you could go through one of the chapter that we wrote, and we enlisted what are the recent technologies has been derived from CRISPR other than genome editing. So we could also forward to you if you want. Just write to me, or maybe my email ID you could get from RSC Bhubaneswar. Also, I can share in the chat box. You could write it to me. I can send it to you.
So hopefully, actually, may I know Mr. Senthil Kumar from US Consulate, Hyderabad, is he here? Hello? Hello?
Sir, I think that Senthil Kumar sir has left because they have some other programs. David sir also left, that's why.
Achha. So Sir Soman sir is there?
Available, sir. So I will give the vote of thanks.
Okay. So you continue now.
Thank you so much, sir. On behalf of Regional Science Center, Bhubaneswar, I myself, Ravindranath Marandi, would like to give the heartful thanks to our Kutubuddin sir for sparing a valuable time to give a very interesting topic on genome editing. And I might have heard about in my class too. I have read about species and genome and how the genome has been editing. A little bit of things has been taught in our Ravensa University colleges. So it was recalling something behind that, how the genome has been, that means technology has been advanced over here. And I think from all the participants that they have got some ideas about why we need to do the genome editing. And hopefully they have got some basic information, like our speaker has been told that all the participants has got the information. So again, from Regional Science Center, it was a great privilege to work with US Consulate General Hyderabad, because this is the second time we are organizing such an international event with our Odisha participants. So thank you also for the US Consulate General Hyderabad. And I will be give a special thanks from our Regional Science Center to our Dr. Srinivas Patnaik sir. Thank you so much, sir. Within a short time, with the communication, you have confirmed us to be a participant. And thank you so much from our bottom of our heart. And I would also like to thank all the participants that the first time we have conducted such a event with all the participants of BSc and MSc in biotechnology field. And again, from US Consulate General and United States India Educational Foundation and Regional Science Center Bhubaneswar, I thanks to all the participants and especially to the Dean School of Biotechnology, Dr. Srinivas Patnaik sir, and say special thanks to our Kutubuddin sir that a long that event has been planned, but finally it was successfully conducted and sparing a valuable time to us. So thank you so much, sir, again, and namaskar. From this, we will conclude today's session. Thank you all.
No, sir, no, sir. Thank you very much.
Thank you, sir.
Thank you.
Thank you.